All right, well, welcome to Biostatistics and Human Genetics. So this is the start of the semester. And just to give you an overview of what we're going to try and get through today is I want to introduce myself and my co-instructors and give you some information about the course and what we're going to try and cover in this course and some of the logistics. And we're going to get through that in a fairly timely manner so that we can get on to starting with the material. And today, for the first time, I'm going to be joined by or for the only time this semester, I'll be joined by my two co-instructors who are also going to deliver some of the lecture today. And so that's the goal for today. And we're actually going to start working with the material in today's lecture. So, as I said, my name is Professor David Gresham. So I'm in the biology department at NYU. My PhD was in human genetics a long time ago. But my doctoral research was focused on disease gene mapping and population genetics. And I'll tell you about some of that research during this course. I actually stepped out of academia for a while, and I worked at a journal known as, which is called Nature Genetics, which is a journal focused on human genetics research. And I was there for a few years at a very interesting time in human genetics. And subsequent to that, I undertook postdoctoral training with a fellow called David Botstein. He, at that time, was at Princeton University and will also, at some stage in this course, encounter David Botstein's contribution to human genetics, which was very seminal at the time. Interestingly, he is a yeast geneticist. That's his day job. He did some human genetics on the side and made some very important contributions. And I trained in his lab and became a yeast geneticist. And so subsequent to that training, I've launched my lab at NYU a number of years ago in 2009. And since then, I've been running a lab where we study a variety of different topics using budding yeast as a model system. We study things like cell quiescence, adaptive evolution, gene expression regulation, and more recently looking at viral evolution and host response. And my lab at NYU, which is at 12 Waverly Place in the Center for Genomics and Systems Biology, employs a lot of the methods that we'll learn about in this course. We do a lot of genetics, and we use a lot of genomics, which is an extension of the foundation of genetics. And we'll learn about some genomics methods in this course as well. And you can go and read more about our research lab at that URL. And we're very lucky this year to have two amazing recitation instructors. So Grace Avicilla is a PhD student. She's on your screen there waving. She's actually a PhD student in my lab, and she studies a class of genetic variation known as copy number variation, and studying that variation in the context of adaptive evolution. And we also have Cassandra Busby, who is in Professor Siegel's lab. She's waving on the screen there at you. And she is a PhD student studying gene-gene interactions, or epistasis. It's a topic we'll learn a bit about in this class as well. And so you have, you're very lucky to have two researchers who are, using the methods and that we're going to learn in this course. Grace has taught this course before, and Cassandra is new this year. And so they're, importantly, both of them also combine experimental research and computational research, and use every day the tools that we're going to learn about in this class. And so let's just start with a very high level question. What is human genetics? What are the questions that we ask in human genetics? And so, human genetics is interested in addressing fundamental questions such as, why do two individuals vary in something as fundamental as human height? Can you see my cursor on the screen? Can I get a virtual thumbs up? Yep. Does anyone know who this individual is here? the very tall person.
Arnold Schwarzenegger.
Former governor Arnold Schwarzenegger, yeah, and Danny DeVito in this really terrible movie Twins that came before your time. But you know, they differ in human height. It's a phenotype that we're very interested in, and we're interested in why do they differ? What's the genetic basis of that difference? So between populations, we can ask, why do individuals differ in both within and between populations? And so we look at these individuals who are of different geographic ancestries and have clear phenotypic differences, and we're interested in what are some of the heritable bases of those differences. Human genetics, of course, wants to ask the question, why does one individual get sick with some heritable disease, whereas other individuals don't? So here, this is Stephen Hawking, who is suffered from ALS, and this is his daughter who was unaffected. And human genetics, one of the fundamental questions we want to ask is nature versus nurture. So what is the contribution of the heritable material that you inherit from your parents to phenotypic variation, to variation in traits and characters? And what is the role of the environment and how do those interact? Okay. And so here we're looking at identical twins. These are actually famous NYU alums. Does anyone know who these two individuals are?
Mary Kate and Ashley Olsen.
Who are were also from the olden times in a TV show. Does anyone know the TV show that they starred in?
Full
Full
House.
House.
Full House, yep, classic. As infants before coming to NYU. So many of you have been exposed to genetics before, and you know, it's an interesting question to ask. What are the differences between human genetics, or what are some of the unique challenges to doing human genetics compared to experimental genetics? So experimental genetics, we can think all the way back to Mendel and studies of peas and phenotypic traits, or all the way to modern day genetics now, where we can do crosses with Drosophila or C. elegans. We can address questions using experimental methods. We can manipulate genomes to address questions. And so what are the challenges that are unique to human genetics? And I'm interested to know what you think. Who can think of a fundamental difference between doing genetics in humans and doing genetics in fruit flies?
Long generation time.
Right. So unfortunately fortunately for us, but humans take a very long time to reproduce, right? So and then they live for a very, very long time, whereas a fly lives for only a few weeks. Or if you work with budding yeast, which your three instructors work with, you know, it has a generation time of an hour and a half. So that makes it really feasible to be able to look at multi-generational inheritance. Whereas for humans, you have to wait a very long time in order to be able to see what the product of a particular mating is. What's another difference with human genetics and experimental genetics?
You can't really manipulate the human genome for ethical reasons.
Correct. So it's very hard to test hypotheses. It's very hard to say, I predict that this particular variant is going to have this phenotypic effect, and then go and test that hypothesis in a human. So that is a challenge. What's another challenge?
Human gene. I mean, there are more chromosomes in human cells than Drosophila cells.
That, and why does that make it a challenge?
There may be more kind of replicates of the same gene in the human cell that if you want to, if modification is possible, it's harder to make sure that the targeted chromosome is edited.
It's true. The human genome itself is complex, much more complex than the yeast genome, for example. Gene structures are complex, and so that does indeed create a bunch of challenges. What about, you know, what Mendel did? Everyone's learned about Mendel, right, back for many years. What did Mendel do? He did a controlled cross. Can you do that in humans? Very difficult, right? You cannot tell two people you must reproduce, and so that we can see what the result of what your offspring are going to look like. That is cannot be done. So you know, we can do that with every experimental organism. We can cross two strains of flies or two strains of worms or yeast, any organism that we're working with, or plants, but we cannot do that with humans. And the other challenge that makes that we're faced with human genetics is that we typically have small sample sizes. We'll revisit Mendel's experiments, but the key thing about Mendel's experiments is that he had hundreds to thousands of progeny that he could analyze. Typical human family in the US, what is it? About 2.2 kids? Is that the average? And so, you know, we have very small sample sizes. There are other problems, challenges we face with humans. One has to do with structure in the human population, that people don't tend to randomly mate. They mate within populations. And so we'll learn about those issues as we go along in this course. And so statistics is really important when it comes to human genetics. So what is statistics? Statistics is the science of collecting, organizing, and interpreting those data. And if we look back through history, human genetics has motivated the development of many statistical methods. And so we're looking at three of the individuals who were most well associated with many statistical tests that we're going to learn about. This is Francis Galton, Carl Pearson, and Ronald Fisher here. They were human geneticists primarily who were interested in asking questions about human inheritance. And in order to address those questions, they developed statistical methods that have subsequently been used in a wide variety of applications. And so I think human genetics is really it's really well suited to learning these statistical methods, because the problems in human genetics have motivated the development of them all as well. We'll learn more about these individuals throughout the course who have made important contributions to genetics and statistics, but also have very problematic historical views that I also hope to address in this course as well. So so you can tell from these gentlemen, they don't look like the most exciting guys. They are sitting in their armchairs and, you know, leading very intellectual lives. And, you know, what they're doing is not really that exciting, because statistics has historically been pretty boring, right? If you had have taken a statistics class in the previous century, which is when I took one, statistics entailed having a textbook that was about this fat, where half of the mass of that textbook was dedicated to having tables such as this, where you'd have to go and look in a table to try and interpret your statistical test. And it was pretty dry and boring. And so luckily you live in the age of the computer, where computing has made statistics, like everything else in life, much more fun and easier. And, you know, there will be challenges along the way, such as Homer's challenge here, looking for the any key. You're going to encounter those challenges as you try and learn about computer programming. You're going to be sitting there wondering why things won't run, but learning is the struggle, is the learning, and you'll learn a lot by doing that. Okay, and eventually you will find that any key. And so in this class, we're going to learn how to use the statistical computer programming language R, which this is their symbol. So computer scientists are notorious for being completely unimaginative when they're naming their programming languages. There are languages C, C+, S, S+, S was the precursor to R. So that is the name of the statistical programming language we'll use. And so why are we going to use it? Well, one of the main reasons we're going to use it is it's free. It's completely free. You can download it from the web and no strings attached. This is in contrast to other languages like MATLAB, where you have to pay a license fee, and that means, you know, when you leave NYU, you don't have access to it anymore. So this is a language you can learn and you'll use for free for the rest of your life. It's also a language that is widely used in the academic research community, and it's also widely used in the commercial world as well. So it's a good language to learn. And finally, because there's this big community behind this programming language, there are lots of people who have written additional tools that we'll learn about that enhance the programming language itself. And so that will be you're going to learn all about R this semester. And so in this course, we're going to aspire to integrate these three topics. We're going to learn about human genetics and genetics in general. We're going to use those problems in genetics to motivate statistics and the statistical methods that we need in order to address those questions. And then to be able to do it in an efficient and reproducible way, we're going to learn computer programming and learn to use the R programming language. And so that's the going to be the goal of this course. And it's all three components are going to be integrated throughout the entire course. So the question is, why learn about human genetics? You know, you might, even if you're not going to go and become a human geneticist and do a PhD in human genetics in your in the future, I still think it's a very worthwhile topic to be learning about at this point. So, and that's because human genetics in the 21st century is going to impact, you know, multiple aspects of our life. So of course it has medicine is one aspect where human genetics and understanding the relationship between genotype and phenotype or disease is going to be already very important. This is Professor Jim Lupsky, who's also an NYU alum. He's a professor at Baylor, but he's been one of the leaders of in human genetics. He's also afflicted with Charcot-Marie-Tooth disease, which is a monogenic disease, and you know he's actually has been instrumental in identifying the genetic basis of the disease that he's afflicted with. Of course, we all know, we've all watched all the terrible TV shows where they use DNA genotyping for capturing criminals. And so it has this, you know, for legal issues, human genetics is very important. More recently, it's become really, there's a lot of evidence in sort of understanding our own personal history through using services like 23andMe. And of course, human genetics and knowledge of the genome and potentially how to manipulate the genome raises all sorts of ethical questions that are new for our generation in many cases. Okay, and this is happening in real time with the advent of CRISPR-Cas9 and the ability to modify genomes. You know, there are reports that individuals have already begun to do this in certain parts of the world, and of course that raises lots of ethical questions. And so human genetics it's a really touches on multiple aspects of our existence. And now is a very exciting time in human genetics. If we look back historically, and we are going to follow a bit of a historical thread in this course, the 20th century, so the century that feels like it just passed, but I guess it was 20 years ago. Maybe some of you weren't even born in the 20th century, is that right? Okay, so in the olden days, you know, it was really an amazing period of discovery for genetics. So it was only in 1900 that Mendel's research was rediscovered, and so we'll learn about that. But it was that just enabled rapid advances throughout the century. So quickly, within the first couple of years, it was the first human Mendelian disorder was identified. A decade later, the physical basis of inheritance and chromosomes and linkage were discovered just up the road in Manhattan at Columbia University. In 1944, DNA was identified as the heritable material, and a decade later, the structure of DNA and the understanding of how it could be replicated and transmitted was identified. In the latter part of the century, we became able to sequence DNA. And it was only in 1980, as we'll learn in this course, so only 40 years ago, was the first time that we were able to map human traits in the human genome using DNA polymorphisms. But within 20 years, we had used that map as the foundation to be able to sequence the entire human genome in 2001. It's interesting that project was, one of the leaders of that project was a professor at MIT, Eric Lander. And I just saw that Eric Lander has been appointed to President Biden's new cabinet. So we have an amazing leader in human genetics as a cabinet member of President Biden's new cabinet. So then with the dawn of the 21st century, things have become, continued to be really exciting. Methods were developed for being able to interrogate the genome using hybridization technology and DNA microarrays. And they lasted for about a decade before new methods for DNA sequencing emerged, which we refer to as next generation sequencing. And this has propelled advances in characterizing genomic variation. And we now live in a world where you can sequence your own genome for around $1,000. And the advances just keep coming. In the last few years, we've had the CRISPR-Cas9 genome editing advances, and continuation in technical advances, being able to sequence individual cells, sequence the RNA in those individual cells. And so, behind this and behind many of the advances in human genetics has been a dramatic decrease over the years in the cost of DNA sequencing. So the first human genome, which was sequenced in 2001, cost $3 billion to sequence. So that was $1 a base on average. Took a huge international effort, but now I can sequence a human genome in my lab tomorrow if I so choose. And so a major driver of these advances has been technology and these DNA sequencing and genotyping methods. And so by the end of this course, you'll become familiar with these methods and how they can be applied to be able to look at DNA sequence and genotype variation, and some of their applications for interrogating gene function in the field of genomics. And so why this course is important is because these advances, these technical advances, have made biology now is a big data discipline. You might have heard of big data. It's the hot topic at the moment, and data science. But in biology now, we generate data sets that are massive, on the scale of 10 to the 9 pieces of information per experiment is pretty standard now. And so in this class, we're going to learn methods that are the foundation of being able to now tackle these questions using big data, the big data sources that are available to us now. So what we will learn in this class will provide a foundation from which you can then continue on to be able to get into higher level statistics, machine learning, AI, which are becoming increasingly relevant for biology. So just to tell you what's not covered in this course. So in this course, there's no molecules, there's no cells. I expect you to know that. You've taken POB, you've taken MCB1, we're not really going to talk about molecules or cells much in this course. What we are going to talk about is 4 main topics. We're going to talk about transmission genetics. So that's how genes are transmitted from one generation to the next. We have a section on population genetics. That's looking at genetic variation within populations. Then we're going to talk about quantitative genetics. So that's quantitative phenotypes and how we understand them. Phenotypes like disease risk for complex disease or human height. And then towards the end, we're going to talk about genomics and get into some of those topics. Just to give you an idea of the structure of the course, so you've all seen the syllabus, but just to explain it, so each week we'll have 2 lectures, and the lectures are designed so that we're covering a topic in genetics and the related topic in statistics. So I've tried to outline that on the syllabus. And so every week it's going to be both the concepts, the genetic concepts, and the statistical concepts that are related to addressing those questions. And every week we're going to have an assignment. That assignment is to be completed in R, and that the instruction for that assignment and the tools for doing that assignment are going to be delivered in the recitations. And so I think I say here, so just to give you more, so just to clarify here, so in the lectures that I deliver on Tuesdays and Thursdays, I'll be talking about the concepts. I'll be talking about the genetic concepts and the statistical concepts, but it's in the recitations that you'll be learning the application of those concepts through computer programming using R. And so the recitations are, to a large extent, almost like a separate course in which you have distinct material that's not going to be covered in the lectures. So it's absolutely critical that you're going to both the lectures and the recitations. I have slides that I'll post before the classes, but the slides are by no means comprehensive, and so you need to attend or watch the lectures in order to be able to get all the material. Okay, but I am going to record everything. I know you're dispersed across the world, and so everything will be recorded and available in the cloud recordings on Zoom. In terms of the assessment, we're going to have we have a weekly quiz on Thursdays. We're still figuring out the best way to implement this. But essentially, this is just going to be on the last 2 lectures. And this is just to compel you to go back and look at those lectures and try and identify the key points. And what most of these quizzes will be on the formula that we have covered in order to be able to do particular statistical tests. We're going to have this weekly assignment. We'll have 1 midterm and we'll have 1 final exam. The midterm and final exam are not using computer programming. They're using pencil and paper, and so to do statistical analyses. And so you're going to learn about that in the course. Okay, and so you refer to the syllabus for all the additional information about office hours and contact details. It's pretty comprehensive, and so let me know if you have any questions about that. There is no textbook for this class. But I am in the process. The NSF has funded me to write a free online textbook. I'm in the process of doing that. And so I'll refer you to the relevant sections on that. It's incomplete at the moment. It's an ongoing project. It turns out to be really difficult to write a textbook. But I'm going to provide you with the relevant sections of that to use. I'm writing this textbook in R, which is, by the end of this course, you'll be able to do too. So you'll be able to write your own theses in R Markdown, which is something you'll learn in this course. Just the assessment, here's the rubric for the assessment. It's primarily the assignments. And so the virtue of these assignments is that they really, it's all, it's very hard to fall behind because it's not like you need to have you're going to have 100% final. So we'll certainly be ensuring that you keep up to date on everything. And then just to reiterate that, we're not, in this class, we're not doing a lot of memorization of facts. It's very much about the concepts. And that's what we're going to be aspiring to have you learn.